episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome, everybody, again to another episode of our show with another really fascinating guest helping to make a better tomorrow on many different fronts. Today, we have the honor of being joined by Dr. Jonathan Chernoff, who is the Chief Scientific Officer, a Senior Vice President, and Deputy Director at the Fox Chase Cancer Center, where he coordinates and charts the future course for research for the organization. And for those less familiar with Fox Chase or the area, it's part of the Temple University Health System nowadays, one of the oldest cancer research and treatment centers in the United States, founded back in 1904. It became the National Cancer Institute's, one of their first institutes designated as a comprehensive cancer center back in 1974. Dr. Chernoff joined the staff in 1991. He was an associate member, promoted to a member of tenure in 1996. In 2002, he's promoted senior member, Fox Chase Cancer Center's Basic Science Division, which is the equivalent of full professor in a university. He's a molecular oncologist as well as a board-certified medical oncologist, and has a specific interest in factors that control cell growth and movement, including oncogenes, tumor-suppressor genes, and has made a lot of fundamental contributions in this area of research. Dr. Chernoff earned his medical degree and his PhD in Mount Sinai School of Medicine, New York City, completed his residency in internal medicine at the University Health Center of Pittsburgh, and a clinical fellowship in medical oncology at Johns Hopkins. Then he did postdoctoral fellow in cellular and developmental biology at Harvard University before he came to Fox Chase. And in recognition of his reputation in both molecular oncology and biochemistry, he also holds the Stanley P. Ryman Chair in Oncology as well. Dr. Jonathan Chernoff, thanks so much for taking the time to come on the show today.
Oh, thanks for having me. I look forward to doing it.
Yeah, it's I very much look forward to talking with you today. You know, I'd really like to start off, as we typically do, by handing you the floor just for a little bit to further talk a little bit about yourself. If you could sort of take us back a bit to you know, where you grew up, how you got interested in medicine, and a little bit of your journey at both the bedside and now back to the bench. I think that'll set the stage for a lot of what we're going to be talking about.
Sure, happy to do so. So I come from a large family. I have 5 brothers and sisters, and in the family history, there's always been a few dominant themes. Medicine is certainly one of them. Poetry is another. Education is a third. And it seems like everybody in the family assorts in every generation along one of those 3 axes, if not multiples of them. You know, more recently, my own dad was a very important influence on me, was a GP. So he was one of the last of the old-fashioned doctors that maybe older listeners may remember, that people used to do house calls. So where I grew up in New Haven, Connecticut, my dad, along with 2 other intrepid physicians, pretty much covered the population of New Haven and did house calls, you know, all over town. And, you know, that style of medicine, for all of its economic flaws, did have a tremendous impact on patients and on you know, people like myself, the children of these doctors, because they were beloved in their communities. I mean, they were going out into the communities, into people's homes. You learned a lot about people that you could never learn from an office visit or a hospital bedside chat. And it's unfortunate that that's been lost in modern medicine. I get the reasons why it happened, but I grew up, you know, in a household where my dad was going on house calls. He would occasionally take one of us along if it was the early evening or something. And you know, you'd sit in the kitchen, typically with the daughter of the family, maybe for some future match that there was being arranged. My dad would go upstairs with his, literally the black bag that the physicians used to carry. In his case, he had a little EKG machine too. He could do cardiograms, portable, which was, you know, state-of-the-art technology in the 60s. And, you know, it was just a whole, almost a romantic era when you look at it back from today's perspective. But it engendered in his children a tremendous admiration for both him as a person and for the field that he was in. And 4 of us out of the 6 became physicians or scientists or both, like in my case. My mother, on the other hand, was an educator. I mentioned that was another strand in the family history. And then later in life, my father died quite young when I was only 15 and he was only 54. And she sort of had to take over the management of 6 children and all the economic issues and so on. That was quite a disastrous time. But she kind of reinvented herself as a poet later in life and published 2 books. She just died 2 weeks ago, unfortunately, at the age 98, but she was lucid until almost the last hour. And her last words were really about getting that 3rd poetry book out. And we promised her, and her editor promised her, that we will do this posthumously, and we are going to do it. So those were my influences growing up. And when I went to school, which was really, college by then, my dad was gone, I was really fascinated by the medical field for all the reasons I just mentioned. But I also loved science, and I was pretty good at it. And I just couldn't decide which to do, medicine or science. So I ended up kind of punting on the decision and doing both for as long as humanly possible. So I did an ND PhD program, which allows you to get a PhD at the same time you're earning a medical degree. And then I did an internship and residency, because I felt like you weren't a real doctor if you didn't do that further training, that just going to medical school gave you the academics, but no practical experience. And then I did an oncology fellowship, because that was my main interest in oncology. And then a postdoc after that at Harvard to firm up my science bona fides, and then eventually joined here at Fox Chase. And even at that time, I was now in 1991, you mentioned, I still couldn't quite decide. And I became, I think, the only person in the history of this place to be both in the science division and in the medical division.
Okay.
We don't have a divisional structure anymore, so it's probably I'm the first and the last to do that. But I think they couldn't quite decide what to do with me either. And so, I joined. I eventually stopped doing patient care. So I like to joke now, my only patients currently have 4 legs and a tail. Usually I'm giving them cancer rather than curing them. But
Sure.
I don't see patients anymore, but in the beginning I did, and I gave it up somewhat reluctantly, but the science part became too overwhelming to do part-time. And I actually didn't feel like, in the spirit of my dad's being a physician, I didn't feel like being a half-time doc was really good enough for the patients. And so my contributions could be more in the laboratory and directing research rather than actually doing patient care myself. And I think about that from time to time, but I think it was the right decision for me.
Outstanding.
That's how I got where I am. It's
No, it's a wonderful journey, and once again, thank you for sharing about your mother. And I'm really sorry about that loss, but it seems like major influences on both sides there from both your mother and
both are very important. That's for most people. Their parents, they're a very important influence.
Absolutely. So you practiced at the bedside. You're at the bench a lot now, but now you have this other major responsibility. And you and I chatted a little before. I could just scratch the surface, sort of the last century of what's happened at Fox Chase. Some of the things that we've talked about on the show: 1952, the first cloning experiments, Briggs and King, Brooke Blumberg, hepatitis V, Nobel Prize, Beatrice Mintz, who we'll get into in a little bit, major seminal work, teratocarcinoma, developmental biology. The list goes on and on. I could probably go for a half hour of everything that's come out of Fox Chase. Talk just a little bit, if you would. I mean, now you are the captain here, steering the research engine. What's this like for you? I mean, I think I'm sure there's things that keep you up at night. You have really a long heritage of stuff that came behind you.
Yeah,
Talk a little bit about that.
I'm up at night usually for good things, like thinking of exciting stuff that we could do. I mean, that is one difference from being a physician, where I'd be up at night because I'm worrying about whether a patient was going to crash or not.
Right.
Now the only tragedy is if a mouse dies and it's not as big a deal, or someone spills reagents on the floor. But so I'm generally up because I'm excited about something that we could try. And one of the great things about being in science is you really are your own boss. I mean, I'm technically the boss of all the scientists here, but I think if you ask them who's their boss, they'd say nobody, or I'm my own boss. Because that is one of the beauties of being in science. For all of the hassles of trying to get funding and keeping the operation going, you drive your own show. And if you think of a great idea last night, you can execute it the next morning. There's very few jobs that give you that kind of freedom. So I've found it the most liberating job and the best job in the world. And for those who are so inclined, I recommend it highly. I mean, you do have to take the bad with the good, but there's a lot of good. And it's just not that many jobs where you are totally dependent on your own thinking. I love it for that. And I think the folks who work here do too. And as you mentioned, some of the greats who came before me, I mean, I occupy the Ryman chair. You mentioned Stanley Ryman. He was our founder back in the early part of the 20th century. And he had some amazing insights, which may seem trivial today, but at the time were not. He thought we could never really understand cancer unless we understood normal cells and normal biology. Now, the opposite is also probably true. You probably can't understand normal without looking at the abnormal. And so studying cancer teaches us a lot about the normal state, but I mean, it goes both ways. And so he insisted on a vision that would study normal biology, which was just not what most cancer people were doing back then. And he also had a lot of other insights about the influence of microenvironment. He didn't call it that, but again, if we get to talk about Min sometime, that was one of her major contributions about what, cancer cells or tumors don't exist in isolation. They're in a body, they're in a place, maybe surrounded by normal cells. What is the influence of that interaction between the cells of the body that aren't cancer and those that are? Fascinating topic in itself, which is still, not that well understood today. So I found it a profound honor to get this chair and to become the scientific director at a place like this, where my predecessors were all, incredible people. Al Knudsen, the guy who hired me, the founder of the so-called two-hit theory, which animates all of our thinking about cancer genetics today. Barry Blumberg, you mentioned, Ernie Rose, who won the Nobel Prize later for his work on protein degradation, Bob Perry for how mRNA works, Ann Skalka, who was my immediate predecessor as scientific director. It's just, a lot of formidable folks, but they drove the recruiting of a great team of their successors, who many of whom are still here to this day. And I hope that my recruiting is as good as theirs, because we have some great young folks here now. I don't know that anyone will win the Nobel Prize, but there's certainly people doing great things today, as there have been throughout the history of this little place.
Absolutely.
I don't do this alone. Of course, I have great people who I talk to all the time, and it's really the conversations like what we're having today I have with others here all the time. And we talk about what is the thing that we should be doing. We're not a gigantic institution. We can't outspend large places. And saying cancer genomics, we are not going to be sequencing every tumor to the nth degree the way that maybe a larger place might. But we can do a lot of things that other places don't do. And so my job is really to find those areas that are not being completely dominated by other groups that we might make a good impact. And a good example is Ernie Rose, one of our two Nobel Prize winners. Here's a guy who was a biochemist, really did all test tube work, was not particularly interested in cancer, I would say. I didn't know him that well, but at the end of his career, I knew him. And but he was fascinated by a topic that others were not so into. He wanted to know what's the secret behind protein degradation, what regulates that. And at the time he was doing this, it was really starting in the 50s and 60s. Everybody else who was a big shot in the area was working on how proteins are made, not how they're destroyed. Right. That was thought to be the interesting question. In fact, the degradation was considered like a housekeeping or almost garbage function. Who cared? Well, he cared. And he plugged away at this for a good long time, publishing in kind of modest journals, I would say, by today's standards. But, good stuff, but not things that were making the headlines in the New York Times. And then he eventually teamed up with the two Israelis, Chechanover and Hershko, who came to work with him in the summers in the late 70s, early 80s. And within about 3 or 4 years, that team cracked the problem of how proteins are degraded. And you might say, well, so what? Maybe the people who thought it was a stupid problem to work on were right. But, that led to an understanding of biology that directly led to the development of drugs like Velcade that's used in myeloma. So that kind of blue sky research, just animated by fascination with the biologic process, right in Stanley Ryman's prediction almost, or his paradigm, led to something more important than most people do in a lifetime in cancer research. So, one of my jobs as a director is to try to recognize, to the extent that one can, which biologic problems are interesting and worth, plugging away at, even if other people aren't saying that they're the greatest thing yet. And, balancing that with more directed cancer research, because this is a cancer center. We're not, just an institute anymore. We treat patients. There is some more applied research, I would say, but also a fair amount of we'll call blue sky research, of just stuff driven by interest. And more great discoveries tend to come out of that. The risk is a little higher because you don't know. Some of these don't end up, with Nobel Prize type discoveries. Some end up somewhere else. But I think Ryman's insight was still right. To understand normal biology tells us a ton about abnormal, including cancer, as well as other things. So that's kind of my philosophy. If I had to articulate one, in practice, it's always difficult to reduce it to, what exactly are you going to do next? And how is that going to make a difference? I have to justify that all the time to, my colleagues and others here.
Excellent. And, thinking about that, understanding the normal before the abnormal, looking specifically now at aside from running the scientific organization, you also have a lab, and you focus on two very interesting components of the transformation process. You focus, as you were saying before, on the signaling, the microenvironment, maybe some of the epigenetics, but there's another part of your research which is equally fascinating, sort of the biophysical aspects of cancer. You look at things like cytoskeletal structure, adhesion molecules, the morphologic transformation. Talk about a little bit, in your lab, how you sort of balance these things, because it's kind of, I don't know if it's chicken and the egg when it comes to a lot of this stuff, but
it is. And I'll probably, go through my entire career not totally understanding this, as these problems end up being pretty deep. And one thing I've noticed in biology in general, and certainly in my own work, is you don't have to scratch too far under the surface before you find something unknown. In fact, for all the things we think we know about cancer and biology in general, I can take any graduate student, and within a week of them digging into some problem, they're going to come into some issue where nobody in the world knows the answer. That's one of the beauties of it. I mean, you can always find interesting things to do, but also tells us how far we've got to go. So yes, I've been interested in the signaling issue ever since I got into this business. And I was fascinated as a graduate student just by these cascades of reactions that occurred, not in a cancer setting, but in glycolysis and how we handle sugar.
Sure. So
when I was a grad student, way back in the day, we didn't have a lot of knowledge of pathways that we do today. But one of the things that had been worked out in the 50s, 60s, and 70s was how glycogen, our storage of sugar, essentially, how that gets stored and how it gets freed up in times of stress, like after you've heard of the fight or flight response. And epinephrine happens and you suddenly need a burst of energy so you can run away from whatever the threat is. Well, that involves breaking down glycogen into glucose, then metabolizing it. And that was sort of the state-of-the-art signaling back when I was a grad student. It rapidly changed afterwards. But I got interested in that signaling process, started studying some of the enzymes that regulate it. And there's chiefly these categories, which I'm sure you know well, but perhaps listeners don't: kinases and phosphatases, enzymes that regulate how phosphates are put on or off of other proteins. And that turns out to be a major regulatory event that governs just so much of cell biology and cell physiology. That actually, discovery of kinases and phosphatases led to a Nobel Prize also. Yep. Back in the 90s for Krebs and Fisher. So I got kind of taken up with that. And then when I did my postdoc, I was with Ray Erickson, who was the guy at Harvard who discovered the activity of the Sark oncogene. So again, going back in the history a little bit, Sark was one of the very first oncogenes to be described in any detail. So, early in the 20th century, Peyton Rouse had done experiments with a chicken virus, an avian virus that caused sarcoma in chickens. It was decimating these poultry populations in New York State somewhere. And so he started filtering extracts from these chickens and ended up defining something that ended up being called Rouse sarcoma virus, named after him. And he actually won the Nobel Prize many, many years later for that discovery. So that was a transforming virus. Ray Erickson, my PhD, my postdoc advisor, figured out that the reason that this virus caused cancer was it contained a gene called Sark, which he then showed with Joan Brugge, one of his postdocs, was a protein kinase. So this lit up the field because suddenly you had this enzyme, which we understood something about. Protein kinases were becoming popular because of the work in glycolysis, et cetera, that was causing acute cancer, at least in an experimental animal, chickens. And very soon thereafter, another scientist named Tony Hunter found that this kinase had a special property. It was that it phosphorylated proteins on tyrosine residues, which, again, may be more detail than you want, but that opened up a whole new class of transforming genes and their activities. And it turned out lots of other viruses contain variants that weren't Sarc, but were kind of relatives of Sarc. So this whole family of tyrosine kinases, many of which were acutely transforming. So I think a lot of people like myself then got really invigorated by this and said, we've got to study these. We kind of know what to look for in a kinase. And now we know it's associated with this acute, cancer-causing activity. And so, people went to town, so to speak, in studying these. And, what did they do? How did they do what they do in cells? And is it relevant in humans? Lots of different questions. And, in terms of the research, when you had an enzyme like that and you knew its activity, one of the chief jobs then was to find what the targets were. What was it hitting that then caused these cancer-causing reactions? And to this day, we're still in that business, but a lot of it has been worked out over the decades. So it got me interested in that field. And, I still, my work today is still an echo of that early stuff. I don't work on Sarc anymore, but I work on things kind of in that same general category. I ended up on my own discovering other kinases that are important in cancer, just they weren't Sarc. That was done already. And I became fascinated with the whole transformation process that was induced by these things. Albeit an artificial setting where you put the gene into a cell, you'd see not only did they start growing differently, but their whole shape was different.
Yep.
They'd start, their surface became very wavy, something called ruffling was occurring. They started drinking up nutrients from the environment, which is an important process, how they feed themselves. They started scattering around the plate in a way that was very reminiscent of, say, a tumor cell in a body metastasizing. And so, a lot of my work in that time has been trying to work out the activity of these kinases and how it relates to a cell proliferating and moving. Sometimes you can separate those out. Sometimes they seem to be very closely aligned. And that's interesting in itself. And a lot of it relates back to developmental biology, because all the sciences are becoming one now in a way.
Sure.
The same processes that drive an embryo to develop and move cells around, like melanocytes, for example, they start off in the neural crest and they end up, in the periphery of the skin, and they take a long, long journey along the way.
Yep.
Well, those processes that govern how melanocytes move are also relevant in cancer, and some of the pathways I study directly impact that. So one of the things that's changed in the whole field is a cancer biologist like myself, if I tilted my work just 5 degrees to the right, people would call me a developmental biologist. 5 degrees to the left, I might be an immunologist. I mean, they're all very, very closely aligned. And I think the categories themselves are almost useless now. We're all just molecular biologists in one way or another. And we share a lot of methods, and the pathways are common. In fact, one of my big discoveries in the early part of my career was just showing how conserved some of these pathways are. So one of the questions back in the early 90s was, if we figured out how proliferation was controlled in one type of system, would it apply to another? That seems obvious now that it would, but back then it was very much a question. So for example, there is a famous pathway that controls proliferation called the MAP kinase pathway.
Sure.
Yeah, you see that the word kinase is in there because kinases govern almost everything. Well, it was worked out best in budding yeast. So the same yeast that you make beer with or bread. And the genetics there were pretty easy to do back at the time, where it was harder in other systems. And they worked out a whole cascade of reactions. There was MAP kinase in the bottom, and there was something above that and something above that. And that was great. I mean, a lot of great papers, but was it relevant to us? And it wasn't at all clear whether that pathway would exist in other types of eukaryotes, like people. Now then, shortly thereafter, someone discovered the same system in the same order was in fission yeast. So that's a yeast that is used in beer in Africa in some places, but it's not typically one that people are familiar with. Well, fission yeast have a similar system. And I think a lot of people then conclude, well, big deal, one yeast has it, another yeast has it. It doesn't say whether it's going to be relevant for us. But the thing to realize is that fission yeast and budding yeast, they share a name. They call it yeast, but that's almost meaningless. I mean, that's like calling something a beast. It just says it's alive. It's a single cell eukaryote. They're about as different from each other as they are from us. So to have this pathway conserved in the two types of yeast, which are 1 billion years apart in evolution, means it very likely that it's common in all eukaryotes. And it turned out to be just so. So in my old lab, the Erickson lab, which had worked on SARC, one of the graduate students at the time named Craig Cruz, who's now at Yale, discovered the activator of MAP kinase. And it turned out to be an enzyme just like the one that was in the two yeasts, called MEK, which is now a target for a lot of therapies in cancer. And I was working, just to get back to my own stuff, in the elements above that. So there's like 4 proteins in a row that are in this pathway. And I discovered a couple of the upper elements, which also had their analogs or homologs in yeast. So not only did it give me something interesting to work on and a new aspect of human cancer biology, but it also really firmed up the notion that what we were learning in these lower organisms was likely to be pertinent in us. And so that, I mean, I didn't make that conclusion on my own. Lots of people in the field, as they developed, began to realize this, but that led to a real renaissance, I think, in model organisms. I mean, for all their weaknesses, and there were many, there's a lot of good things that you can learn from other systems. And we've learned a phenomenal amount in these more tractable systems. Now, today, the game has kind of equalized because of CRISPR and other methods. We can do genetics just as easily in human cells as we can do in yeast. Back then, that was not the case. It was much easier to work in yeast. And so now there's a trend a little bit away from model organisms. And there's also other complex interactions that you can't model well, like tumor environment, that just is harder to do in those systems. But I would say we learned a ton from, I mean the field and myself, from working in model systems. And now it's kind of a mishmash. People do some there and some in mice. Some people don't like mouse models for various reasons. Some experiments are not really done in human, but we can infer things from patients now that can add to that knowledge base. So it's been fascinating just to watch the evolution of how people even do their experiments, let alone what the content is.
Absolutely. Jonathan, and I encourage everyone watching and listening to go into PubMed and search on Dr. Chernoff's work, and you have a long list of publications and lots of these targets that you've published on. Thinking about transitioning to sort of the molecular therapeutics part of Fox Chase now, and obviously the interest to move these findings into the clinical setting at some point. We've talked a lot on the show about sort of history, from the cytotoxic era to now sort of the smart drug era with the antibodies and the CAR T cells and so forth. I'd love your take on sort of where we're going in terms of these drug development trends. I I talked to Azareza a couple months ago. She's very hot on getting the early cells. Combinations are always been something that is very hot, obviously, in oncology.
Right. So those are great issues. It's what I think about all the time. And, anybody worth their salt has to think about this in this business, because, let's take a reality check on where we are in clinical cancer care. It's not way different than when I was a fellow 30 years ago, unfortunately. So if you're not cured early, like detected and cured, and a lot of people are, it's more than half of cancer patients get cured right up front because whatever they have is small enough that can be excised or radiated or something. Or for pediatric case, it's different. Children commonly cured of their cancers. But for adults who have, solid organ tumors, pancreas, lung, liver, etc., we don't have anything radically better than what we had before. There are definitely incremental improvements, and I don't want to minimize those. In fact, I'll talk about them in a minute, but we have not cured these things to any great extent. And even in the liquid tumors like acute myelogenous leukemia, we're not way better than we were. It's incrementally better. And that's despite targeted therapy. So you mentioned, these sort of magic bullet type things. Well, that has been the pipe dream of oncology for the last 25, 30 years. So as soon as sequencing became a thing that you could do relatively easily, people have applied it to the tumor genome, because I think most people agree that cancer is a mutational disease. It's a disease of mutation of genes, various either tumor suppressors or oncogenes, etc. And so the idea was born that if we could figure out the signature, what genes were mutated, then we could match up people with therapies. And there was a fantastic example, maybe too good an example, 20 years ago with the BCR-ABL gene in chronic myelogenous leukemia. And this actually has a big phosphatase connection because the Philadelphia chromosome, which is the chromosome that is found in 95% of people with this particular form of leukemia, was discovered right here on this campus by Peter Knoll, who was a UPenn person, and David Hungerford from Fox Chase. So I actually have the microscope. It's not one of the ones behind me here, but it's on display. I got it from Hungerford's widow. But that was done here, and it was the first chromosomal aberration seen in cancer. Now, fast forward 20 years, people figured out what the meaning of this Philadelphia chromosome was, what was at that junction in this abnormal chromosome. And it turned out to be a tyrosine kinase, a gene we've been talking about, called ABL, and it fused to another protein called BCR. So BCR-ABL was the oncogene driving chronic myelogenous leukemia. That led people to say, okay, now I've defined the lesion. And now if I could only make something that would target that, voila. And that's exactly what happened. It was an incredible success story. There was a compound already out there called Gleevec, and Brian Drucker and others started testing this, figured out it worked extremely well, both in experimental settings and then eventually in people. So by 2001, I think that got approved. It was one of the first of these kinase inhibitors to make a difference. And I said before, it's almost too good an example because it fooled people into believing that we would have equivalent success in other settings. And that has been not the case, unfortunately, because CML, this leukemia, is a very simple kind of disease. It's driven by a single genetic lesion, one mutation causing these cells to go off from being normal into this leukemic state. One therapy. Now, even those therapies, resistance did develop in time, but they had backup systems where they could then take care of the kind of resistant cells. So it turned out to be a very tractable problem. So people with CML today, unlike when I was a resident, don't end up generally needing bone marrow transplant. They can be controlled very nicely by taking a pill every day and live a normal life and die from something else. And so that is a great paradigm. I wish that we could do the same in all diseases. It turns out there's a minority of cases that are like that, other diseases where a single mutation is responsible for most of the bad things that happen and that we can target and cure.
Right.
Much more common is that there's a whole bevy of mutations, many of which we can't target today, or even if we can, resistance develops very rapidly. So a good case study in this is in melanoma. 20 years ago, they discovered that the BRAF, another kinase, was mutated in half of people with melanoma. Fantastic finding. And by the paradigm that we discussed for the people with leukemia, say, okay, let me target this BRAF protein, and that should be enough. And so that was done. A very effective compound that's still used today called vemurafenib was made, and others like it. And there were unbelievable results in that you would take a patient who had, floridly metastatic melanoma, like on their skin, on their chest, everywhere, treat them, and within a few weeks, it all melted away. Beautiful. But then the almost universal experience was a few weeks later, they would all come roaring back again. And that is unfortunately the more common scenario in most cancers. We might have a targeted therapy that we can nominate based on your particular mutation, but our success in cure in most cases is very low. And so most of what I do today and what I am trying to push here as an institution is to deal with the drug resistance problem. So we're great at diagnosing these things in terms of their molecular defects. We can do that. Any competent cancer center can do that today. We're less great as a center and as a world effort at dealing with what happens when our best therapies fail, which they do. So there we have to figure out, well, what can we do in addition? And so if this BRAF drug was so great in melanoma up front, that's nice, but can we figure out what to do next or what to combine it with? Now, in the case of melanoma, there is some hope because in an entirely different type of therapeutic, namely immunotherapy, has turned out to be quite useful in melanoma, both on its own and in combination with these type of targeted therapies that we've been discussing. So one of the common regimens now, again, at a cancer center like ours or others, is to combine a BRAF inhibitor, if you have that mutation, like in half the patients, plus some kind of immune checkpoint therapy. Now, even that is not necessarily curative. There's plenty of people who fail, and there's a lot of research. And actually, I shouldn't say that people fail. It's the drugs that fail. The people have nothing, it's not their fault. But the drugs fail, and these combinations fail more often than we would like. And so, we're looking, what else could we combine? And so, in thinking about this general problem of drug resistance, we've thought of a bit different approach than most places. So what most places will do is they will look hard at the genomics. They will ask, well, did additional mutations acquired? You can imagine under selection of some agent, like if you were a bacteria and I gave you penicillin, if one bacterium figured out a mutation that would resist penicillin, soon enough the entire population would be resistant. And that happens, in infectious disease. Cancer is not that different. If you treat with something that doesn't completely wipe out the population of stem cells, and one of them acquires a way around your drug, then that will end up dominating the population later. So you will have a sort of a worse version after treatment that's more resistant than it was before, almost by definition. And so I think one of the insights or one of the approaches we're trying to do here is not focus so hard on the genetics. We do that routinely, but I'm not sure the answer is going to lie there. We have some technologies that we work on, and not totally unique to Fox-Chase, but they're not universally used, that look more on the protein side, like what's going on with your signaling molecules. Can we measure their activities en masse? And the answer is we think we can, and that knowing that might make a difference. So for example, I'm going to compare this a little bit to a chess game. If I'm playing an average player in chess and I open with the Spanish opening, there's about 3 or 4 kind of responses that most adequate players might respond to that. And then, the opening game is pretty well established if you study them, and there's kind of moves that you make, and what your opponent, there's a small menu of things that they're going to do. And that's versus playing like Bobby Fischer or Kasparov or whoever the current champion is. Sure. You might open with that, and they might have 1,000 variations that they can throw at you because they're master chess players. Well, it turns out when we look at cancer cells and we treat them with something which they then become resistant to, they don't have an infinite number of strategies that they use to overcome the initial therapy. They have maybe 3 or 4. So they're more like the high school chess player than Bobby Fischer. Okay. That's good for us, because if they were like Bobby Fischer, we probably going to have to throw up our hands and just say we're going to be defeated. But, I can probably beat the high school player. And if we can anticipate the 3 or 4 things that the cancer cell is going to do, and we're lucky enough to have therapeutics that will deal with them, which in most cases we do today, it gives us some optimism that we can deal with this resistance issue. And it's something we would not see by looking at the genetics per se. It's not necessarily new mutations, it's just new use of existing enzymes. And the reason that there's no one place that dominates in this space now, they can read about it more in the New York Times or other places, is that there's no one dominant technology yet. In sequencing, it's all digital. It's like running a computer. You have 4 bases, so there's basically 4 values, and that is very inherently digital. And as long as you have a way to read them, it becomes more of an informatics problem of people, you don't have to know much biology once you have the sequence. Maybe that's a little too strong to say, but on the protein side, we don't have one universal approach yet. Some people use antibodies to look at these things. Some people use mass spectrometry. We have our kind of version of this thing. And so people are out there, there's a lot of creativity in trying to figure out how can we look at the proteome or the kinome or the metabolome, all these other omes that aren't typically the ones looked at big cancer centers, and use that information to improve therapy. And I don't have a final answer on this yet, but it's in the Fox-Chase tradition of doing things a little different than what other places are doing. That's what I'm pushing as a scientific director now, and I've not encountered a ton of resistance. In fact, I tend to hire people who are in this area, and we have a big effort going on here now, which I hope that we can brag about in a few years when we have some big successes. But it's a tough problem. I mean, overall, this is not easy, or else we would have fixed it already. You may remember some years ago, there was a big headlines again, and I keep mentioning New York Times, but they literally did make the front page. I remember reading it, that the inhibition of blood vessel growth was going to be the answer to cancer. They even had quotes from Watson and others about this was the end of cancer kind of as a problem. And we were joking around here that we would have to change from being the Fox Chase Cancer Center to the Fox Chase Center for Aging or something like that. Well, I wish that had come true, but it didn't. And 20 years later, we're still struggling with this issue. So that, is one more part of the armamentarium, but was hardly what was said. And that's the problem in this field in general, is there's just too much hype. I get it, as a person who's had cancer in our family too, you want to believe and you have to believe, but there's a lot of loss in this field, and you have to, we just aren't quite there yet. It's not for lack of people trying or lack of intelligence or even lack of money. Of course, we prefer having more research money, but it's an authentically difficult problem. And I think it is soluble. We've made a lot of incremental progress, which turns out to matter because, people are always looking for the cure. They want, like in CML, where you take Gleevec and, for all intents and purposes, although you can't not stop taking the drug, but you're kind of safe. In other areas like breast cancer, we haven't had a home run like that. I mean, there's been a lot of things like the HER2 therapies or the antibodies against this common oncogene that's amplified in breast cancer and ovarian. It's made a difference, but it hasn't made that much difference really. But that in combination with things like tamoxifen, better diagnostics, and better supportive care ends up meaning that people who have breast cancer survive way longer than they used to.
Sure.
So enough incremental advances end up being almost like the equivalent of a revolutionary advance. That's happened on a lot of fronts. And it's almost like we have to change our thinking. Are we going to cure things or are we going to manage them? In other fields of medicine like diabetes, it's kind of accepted, at least for now, that if you have type 2 diabetes or type 1 diabetes, it's really not a cure. I mean, yeah, for type 1, it could be a transplant, but, kind of exotic cures, I would say. For most people, it's a disease that you manage. You take various pills, you alter your diet, you do changes, and you can live a long, long time with some of those things. In cancer, we haven't had that concept for as long, but it's beginning to look that way. There's certain kinds of cancers that maybe you can just live with. Prostate cancer is an example of that, where a lot of them are pretty indolent, and it's not clear whether any therapy is really required. Breast cancer, you do need therapy, but some of them can be managed for decades now. And so, that's not as good as a cure, but it's not bad, especially if the therapy is not too toxic, like tamoxifen or things like that. And maybe, then you have other problems to deal with, but at least they're not cancer. So I think we would love to have the revolutionary cures, but enough incremental is pretty good as well.
Sure.
And to get to that stage of, whatever the future is going to hold, we are moving into a new era of genetic medicine where we actually have the technology to go in and fix that which is broken.
Yep.
And so, this is a real frontier area, but we do have the technical means to fix things, and certainly the ideas out there. Why are we using small molecules and antibodies? Why don't we just go in and fix the problem at the source? And that is being done in a limited sense, not for cancer so much as in deficiency diseases like various enzyme deficiencies or things like sickle cell, where you might be able to fix a mutation. And that's been done, actually. There's some trials going on now which have had very interesting and compelling results. In cancer, eventually that era will come to us. And it's already nibbling sort of at the edges of the research world. But you again are facing a way more difficult problem. You're not trying to just replace one enzyme. You're trying to fix maybe billions of or millions of copies of a damaged gene, and how you're going to get that there without messing up other things. It is the next era, sort of the post small molecule era, but it's going to take a long time to be implemented. And it may be my successor who governs research on that. I mean, we are in that area too. I would say, not dabbling is probably too weak a word, but we are doing things, but I don't expect them to make a difference in patients anytime real soon, at least from what I see here.
Sure.
Others may disagree, but it's a new technology. It's just, that could be a revolutionary change that totally alters how we think about cancer, but just not for 2021.
Got it. Let's go back in time for a minute because I'm going to talk about someone who we both really admire, Beatrice Mintz. Let's go back to 1975. Normal genetically mosaic mice produced from malignant teratocarcinoma cells. Major, needless to say, seminal work. And thinking about your chessboard analogy again, about okay, we have these these cancer cells that have a limited amount of stuff that they can evade, and then thinking about a developing embryo or similar dynamics in other systems, which has we don't know, but thousands of enzymes and proteins and microRNAs and other stuff that is going on as that embryo becomes that normal mouse or whatever at the end of the day. And thinking back about some of that work, obviously it laid the basis for all sorts of important developmental biology stuff and disease models and embryological development. But she showed, you could take these nasty cells and turn them back into normal stuff. Why has that? Because that seems to go, I mean, there's stuff that was done before that in frogs going back to the 40s in terms of regeneration. Where are we, or why hasn't the whole concept of tumor reversion, which we know exists, taking something malignant and boom, it's normal again, whether it's in an embryo, wherever. Where does that sort of sit in the
that is
stuff?
one of the, actually, if I had to say a second front besides these sort of proteome analysis that I discussed before that we're pushing at Fox Chase, is the field of epigenetics, which is really the one that would deal with the issue that you raised. So, B. Mintz, who was and is one of the most incredible people you ever meet. She's still alive at age 100 and technically is still on staff here. Was a great friend of mine. And, I spent many interesting hours in discussion with her, and they literally were hours, because once you got in the conversation with B. Mintz, you weren't getting out of that office for the rest of the afternoon, for whether you wanted to or not. And usually you didn't want to, because she had so many fascinating things to say. But this is a woman who worked virtually alone for her entire career. She was a little bit like Barbara McClintock, who was a Nobel Prize winner before, and I think she was friends with her. But she just came into the field at a time when it was very difficult for women. I mean, people talk about today and the barriers that women face. Think about the 50s when she came here, and I think again, my predecessor ×3 was the one who hired her. Just saw that she had this phenomenal talent, convinced her to come here from where she was, I think Iowa, where she was before. She only reached here when she was age 40. So she worked here for 60 years, which is on record for anybody, but she didn't start here as a 25-year-old either. But anyway, she virtually worked alone her entire career. She only had 2 postdocs, and both of them became famous people in their own. So I think her standards were so high. This was Rudy Yanish at MIT and Michael Caron at UCSD. So that was, she was going to get a postdoc. They were going to be damn good, but she didn't have too many in this long, long career of hers. And so she just did stuff according to whatever interested her. And she had many quotes that I can remember, but one is, why waste your time on small problems, was one of her favorite sayings. And I think a lot of accomplished people have that same attitude. Like, you can work on a lot of things hard. Why not work on something that matters? Yep. And so she was fascinated by this whole embryology angle, and she probably should have won the Nobel Prize, but she didn't. But her work was still in that category. I mean, she developed transgenic mice, which we can leave for another time. But she also had this observation that you alluded to, that you could take stem cells that were clearly cancerous, kind of mix them up with normal stem cells in a mouse embryo, and the embryo would come out normal and would go on to normally breed and produce normal offspring. And the way she used to describe it to me was, this is a mouse whose father was a tumor. Yep. And that's kind of mind-blowing even today. As you mentioned, how did they get normalized? I mean, something was dreadfully wrong with them, right? And and she didn't know the genetic lesion, because back then you didn't have the technology, but we knew they were cancerous, the initial cells. And we knew that whatever came out from these mice was normal as far as you could tell. I mean, you couldn't sequence them, but they lived a normal life. They didn't get tumors. They had children. They, behaved normally. So for all intents and purposes, we're a normal mouse, as if they never had this tumor cell father. Yep. And really, that kind of got into 2 huge areas. One is microenvironment and tumor interactions. Yep. So what is it that's being communicated by cells around the tumor that is influencing what the tumor is doing? And conversely, what is the tumor saying to the microenvironment? There's kind of an exchange of information going on. What is that? And how could it be that these normal cells on the outside could make the tumor cell kind of behave itself? And some of the answer to that is influence on what we call epigenetics, which is somewhere in between the two fields we were discussing before, genetics and proteomics. In between them is this big field of epigenetics, which is not mutations. It is alterations of the DNA, sometimes by phosphorylation, the thing I am interested in, or methylation or other modifications, and sometimes modifications on histones, which are the little protein clusters that DNA gets wrapped around in the nucleus. So it turns out epigenetics, and some people might define it even more broadly, but generally modifications of DNA, non-heritable, and modifications on histones, which is what the DNA is wrapped around. And it turns out that has a huge impact on how mutations even get expressed. So you might have a mutant gene, and maybe if it's expressed, it causes cancer or wreaks some kind of havoc in cells. But if it never gets expressed in the first place, then it doesn't do anything. So if you can silence a gene, and I mean, the molecular explanation for some of these phenomena that she observed are that certain genes that would have caused tumors were being silenced. And it may be more than one. I don't think it's ever been completely worked out, in fact, in that particular case, because no one has gone back and redone that experiment exactly as she did it. But her observation was required for the rest of the field to even get going and be interested in this phenomenon. And so there is a big effort in controlling the epigenome. So, and we have a big program in that. In fact, it's one of the few cancer centers in the country that has a program devoted to cancer epigenetics. And that's because in my mind and in my colleagues' mind, this is an important enough field that it needs its own program. It's not genetics, it's not proteomics, it's epigenetics. And it turns out many of the enzymes that are involved in altering the epigenome are druggable. There's tons of interest from pharmaceutical industry now in altering the activities of these enzymes. And so we're working with some of those companies, or we're doing our blue sky research on how this whole system works. It's one of these frontier areas. I don't claim that we're the only people working in it, far from it, but we definitely have special interest in this area and devoted a lot of our resources to recruiting top level talent and putting other institutional monies behind this. So I hope one of my legacies will be to have built up on the observations of B-Mints, but taking it to the next level and taking it into patients, which is something she tried to do near the end of her career. She actually decided at age like 75, I remember her telling me, it's time for me to cure cancer. Okay, well, if someone's going to do it, she'd had enough of developmental biology. And she got into melanoma field. And she was doing some fascinating experiments, different than what I would do, but she really, molecular biology was just a tool for her. It wasn't her whole approach. She was transplanting, grafts of skin from one mouse to another and asking whether the environment of the skin from the normal mouse could influence the cancer, and it turned out it did. At the margins of these tumors, all kinds of things was going on because of communication between the normal cells on the left and the tumor cells on the right. And so, and she didn't end up completing those experiments just because they take forever, and she got older and older. But it was I think a lot of that work will be considered pioneering also in the future as people kind of seize on those observations of hers.
Yeah, absolutely.
But yeah, she was, one of my scientific heroines here. There were others too, but I mean, this place just has so many. Many are like ghosts now, kind of floating around, and you can almost talk to them. But in her case, she's still with us. So,
yeah, really fascinating. Just Joe, one more thing while I have you. You have a lot going on, but I really just wanted to ask you about one other thing. You published at the end of 2019 in Molecular Biology of the Cell journal. The paper was entitled "How Much Is My Paper Worth?"
Yeah.
And
you read that paper.
So I love it because you go into the fact of how much time and energy and money scientists put into creating these scientific publications. And you come back to discussion. What's this all about? You pointed out in the paper the Rose Nobel Prize on the proteasome ubiquitin, that whole story. It wasn't in a major paper. You talk about CRISPR. That wasn't in a major publication. Talk about why you wrote this article.
Well, it gets back to this comment of B. Mintz's about, working on important problems. You have to pick a problem that interests you, whether the rest of the world thinks anything of it or not hardly matters. I mean, if you're convinced that you're onto something interesting, you'll follow it. And, if you do that, a lot of times in the beginning, people are not so accepting or so excited about your discovery. So the initial CRISPR work was done in bacteria, way back when. It was an weird observation of these funny sequences in bacteria. Ernie Rose's long plod through the proteasome work, was of some interest to biochemists, but it wasn't a major interest. And I think he had to have kind of a thick skin. He just have to not give a damn what other people think. That's why you become a scientist anyway, to be an independent thinker. And so, you certainly pay attention to literature and see what's out there. But if you're convinced you're onto something, you got to follow your nose in this. And even my predecessor in this job, Stanley Ryman, who's not really a basic scientist, he was a pathologist. He wrote a very interesting article, which I recommend to you, when he was president of the American Association for Cancer Research many years ago, called The Highways and Byways of Cancer Research. And in it, he argued that there's a lot of people on the highways, but there's a lot of interesting byways. And often the byways become highways in the future as people start to realize what a valuable path they show. And he gave some examples, from history of that. It's kind of like a older version of what I wrote, the article you read. So, most papers are never referenced again, or have, one reference, because a lot of people work on stuff that ends up going nowhere. And that's about half the papers out there, unfortunately. So you could say what a waste, but I maybe we'll have to comment about how science is run in general to get into that. But other ones sometimes take a long time to catch fire. They'll be hanging out there for a while, like Ernie Rose's early papers. They were cited modestly. As soon as they cracked the system and people started to realize how important this was in cancer, suddenly they became citation classics, with thousands of references. So, sometimes you have to be patient, but you really have to trust your gut on these things. If you think you're into something, devote your heart and soul to it. Sometimes, you may be chasing something. You have to be wise enough to realize when you're going down a dead end. But like I said, those byways are only byways because not a lot of people are on there with you, but they may become highways. Yep. If what you find is important enough.
Absolutely. It's
great. So it does take kind of thick skin, and it's hard. Nobody likes getting rejection letters, and nobody likes hearing that you're doing boring stuff. You have to kind of shut all that out, and usually you prevail in that circumstance. I mean, that's why my main thing in hiring people isn't really what they do in particular. It's how creative and talented do I think they are. Sure. If they've got that spark, I am willing to give them a lot of leeway. I'm not so genius that I can figure out everything that's going to work or every trend that's going to be important. Otherwise, I'd have my own Nobel Prize now. But I'm pretty good at recognizing talent, and I will give people enough leeway to do their thing. And like I said at the beginning, no scientist really even has a boss. If they wanted a boss, they'd be in a different type of job. So I can encourage them, I can coax them, I can offer them some carrots. Maybe I can hit them with a stick if they really are misbehaving. But in general, they do what their mind is telling them to do. So it all comes down to their creativity. And in that sense, and to link it to the very beginning of this podcast, I mentioned my family, there were poets and writers as well as scientists. I don't think that they're on polar opposite sides of the moon. We're all explorers here. And if you look at some of my maps behind me, these are exploratory maps. They're about, the voyages of the Odyssey and so on. Because I think, in a way, and maybe it's too romantic a way to think of it, is we are the descendants of these folks. We're explorers too. Mm-hmm. We're poets. We're just working in a different medium. And, what you discover is not going to be something that you wrote out of some book. You have to find it yourself. Yeah. And if you get smart people who are creative and have the energy and drive, good things will happen. That's been my experience.
Excellent message. Fascinating journey, Jonathan, and I just I really wishing you the best with all this, leading the organization forward on this future course of research, and really, as I said, wishing you the best for everybody that's going to be listening to this particular episode on the podcast or watching on the YouTube channel. You've been spending time with Dr. Jonathan Chernoff, Senior Vice President, Deputy Director, and Chief Scientific Officer, Fox Chase Cancer Center, Stanley P. Ryman Chair in Oncology. I strongly encourage everyone to PubMed search him, Google search him, and all his work. Jonathan, I once again take the time out of your schedule. Thank you very much. Thank you for everything you're doing there at Fox Chase. And as we like to say on our show, thank you for helping to create a better tomorrow through all your work. Really very inspiring.
Well, thank you. It's been a great pleasure.